Our bodies are pretty incredible, Like they know how to do so much. Grow, heal, fight off all those nasty bugs. But how do they know when to do what?
Yeah, it really is remarkable, isn't it?
It is.
Well, a lot of it comes down to our genes.
Oh, okay, so like our genes are the instruction manual.
Exactly. But it's not as simple as just having the right genes. It's about which ones are turned on at any given time. That's where gene expression analysis comes in.
So it's less about the instructions themselves and more about which ones we are choosing to use at any given time.
Precisely.
Okay, so then how do scientists actually see which genes are turned on? What tools do they have to peek into this microscopic world?
Well, they've got a few really amazing tools. One of the most important is something called quantitative PCR, or qPCR for short.
qPCR. What's that?
Imagine you need a specific recipe, but you've got millions to sift through. qPCR is like a super-powered search engine for our genes, finding the exact instructions we need.
So it's like Google, but for ourselves.
Exactly.
That's wild. Okay, so how does it actually work? Does it involve tiny robots searching through DNA strands or something?
Not quite robots, but the process is pretty fascinating. First, scientists extract RNA from the cells. Think of RNA like a copy of the DNA instructions the cell is actively using.
So it's like a snapshot of what the cell is working on at that exact moment.
Yeah, a snapshot of the active instructions. Then they convert that RNA into DNA, which is a little more stable, easier to work with.
Okay, so translate the instructions into a language the machines understand.
Exactly. And once they have that DNA, they can make tons of copies of it, millions even. This amplification process makes it way easier to detect and quantify the target DNA.
So more copies means a stronger signal, right? Makes it easier to spot.
Exactly. And because of this amplification, qPCR is super sensitive, like crazy sensitive. It can detect even tiny amounts of a specific gene's instructions. That's why it's so valuable for things like, measuring how genes respond to different medicines or even detecting viruses.
So qPCR helps us see how genes are reacting to their environment. Pretty cool. But what about the actual proteins being made? How do we know those instructions are actually being followed?
Great question. That's where Western blotting steps in. While qPCR focuses on the blueprint, the instructions, Western blotting is all about checking if the building, or in this case the protein, was built correctly.
So it's like quality control for ourselves, making sure the blueprints were actually followed.
Precisely. Imagine taking a toolbox and organizing all the tools by size. That's what Western blotting does with proteins. It separates them based on their size using a technique called gel electrophoresis.
So we have all these proteins lined up, ready for inspection. But how do we find the one we're looking for, the protein we want to investigate?
For that, scientists use antibodies. Antibodies are kind of like these little molecular homing missiles. They're designed to bind specifically to the protein of interest.
So each antibody is like a custom-made tracker looking for its specific protein target. And because we already have the proteins all sorted by size, we can quickly see if the protein we're looking for is there and if it's the right size, right?
Right. And we can see if there are any modifications to it. Modifications to a protein can change how it functions, so that's important information to have. Western blotting is a powerful tool because it confirms whether a gene's instructions were actually followed and turned into a protein. It even helps us understand how that protein might have been modified along the way.
Wow, it really is like having a window into the inner workings of our cells. So we've got qPCR to see the instructions and Western blotting to check if those instructions are being followed. What other tools are in the scientist's toolbox?
Oh, there's also ELISA.
ELISA, okay. And what does that stand for? I'm guessing it's another one of those scientific acronyms.
It stands for Enzyme-Linked Immunosorbent Assay. Sounds complicated, but ELISA is all about precision. It's the ultimate quantifier. It can tell us exactly how much of a specific molecule is present, whether it's a protein, an antibody, or even a hormone.
So it's not just saying, yep, it's there. It's actually measuring the amount.
Yeah, like taking inventory of our cellular toolbox.
That's pretty handy. How does it manage to do that? What's the magic behind ELISA?
It all comes down to the interaction between antibodies and their targets. Think of it like a lock and key, where the antibody is the lock and the target molecule is the key. Only the right key fits the lock, right?
Right. That's how we make sure only the right things get in. So how does this lock and key idea apply to ELISA? Are we setting traps for these molecules?
In a way, yes. Imagine we have an antibody attached to a surface, like setting a trap. If the target molecule is in the sample, it'll bind to that antibody. Then we introduce a second antibody that also recognizes the target molecule, but this one has an enzyme attached to it. The enzyme is like a little beacon that sends out a signal when it finds its target.
So the more target molecules present, the stronger the signal from these enzyme beacons.
You got it. And by measuring the intensity of the signal, we can figure out exactly how much of the target molecule is there.
That's incredible. So we've got qPCR for reading the instructions, Western blotting to check if the instructions were followed correctly, and now ELISA to precisely measure specific molecules. Scientists really have a whole arsenal of tools to explore this hidden world, don't they?
They do. It's amazing what we can learn by studying these tiny molecules and the intricate ways they interact. It's like piecing together a giant puzzle, one experiment at a time.
It really makes you appreciate the complexity of life, doesn't it? All those processes happening inside us right now, all those tiny molecules working together to keep us going.
Absolutely. And the more we learn, the more we realize just how much we still don't know. But that's what makes it so exciting. There's always something new to discover. And each of these techniques, they each give us a slightly different perspective on what's happening inside our cells.
Right. Yeah. Because like we were saying earlier, it's not just about knowing what genes we have. It's about understanding which ones are being used, how much, when, and where.
Exactly. So, for instance, qPCR, that's great for telling us which genes are being transcribed, like which blueprints are being pulled off the shelf, but it doesn't tell us if those blueprints are actually being used to build anything.
Right. Like having a recipe book doesn't mean you've actually baked a cake. We need to see the finished product.
Precisely. And that's where Western blotting comes in, remember. It's like checking to see if the chef actually followed the recipe. We can see if the protein, the final product, was actually made.
And then ELISA adds yet another layer of detail. It's not just about whether a protein was made, but how much of it was made. Like, are we talking a cupcake or a 3-tiered wedding cake?
Perfect analogy. So yeah, using all three of these techniques together, scientists can really start to understand what's happening at a molecular level. But of course, it's not always easy. There are challenges, right?
Oh, I'm sure there are. I mean, this is cutting-edge science we're talking about. What are some of the hurdles scientists face when using these techniques?
Well, one of the biggest challenges is making sure you have good samples to work with. If the RNA or the protein is damaged or contaminated, the results can be completely off.
Oh, yeah. Makes sense. Garbage in, garbage out, right?
Exactly.
Yeah.
So sample quality, that's crucial. But even with a perfect sample, these techniques can be finicky.
In what way?
Well, each technique, it requires really specific conditions to work properly. It's like baking,
Yeah.
If you don't get the oven temperature just right or you use the wrong amount of flour, your cake's going to be a disaster.
So there's a lot of troubleshooting involved, a lot of trial and error.
Oh, absolutely. Scientists spend a lot of time optimizing these techniques to make sure they're getting reliable data. And even then, interpreting the data can be tricky. You really have to be careful, think critically about what the results actually mean.
It's like being a detective, trying to piece together clues. So what are some of the things scientists are hoping to learn by using these gene expression techniques? What mysteries are they trying to solve?
Well, one really exciting area is single-cell analysis. Traditionally, techniques like qPCR and ELISA, they measure gene expression in bulk samples, like thousands or even millions of cells at once, which means you're only getting an average picture.
Right. It's like knowing the average height of everyone in a room, but not being able to tell who the tallest person is.
Exactly. But with single-cell analysis, you can zoom in on individual cells and see what's happening at a much finer level. It's a game changer, really.
So it's like realizing that even though everyone in a room might be wearing the same uniform, they're not necessarily all doing the same thing.
Exactly. And this variation between cells, it plays a huge role in all sorts of biological processes like development, disease, and even the immune response. So yeah, single-cell analysis, that's a really hot area of research right now.
So we're getting a more personalized picture of what's going on in each cell. What other cutting-edge techniques are out there? Are there any new tools in the scientist's toolbox that are really pushing the boundaries?
There are. One particularly cool one is called spatial transcriptomics. This allows us to measure gene expression in intact tissues, but also to see where each gene is being expressed, like its location within the tissue.
Wait, so we can actually see where each gene is located within a tissue. It's like adding GPS coordinates to those blueprints, showing exactly where in the building each piece goes.
Yeah, it's kind of like that. And knowing the location of gene expression, it's crucial for understanding how cells communicate with each other and how tissues are organized, particularly in complex tissues like the brain.
This is incredible. So we're really moving beyond just knowing which genes are turned on or off. We're starting to map them out in space and time.
We are. And as these technologies continue to evolve, they're going to give us an even deeper understanding of how life works, at a fundamental level. And that understanding, it's going to open up all sorts of new possibilities.
It's exciting to think about the potential, isn't it? Like, how could these discoveries actually translate into real-world applications? How could they impact our lives?
Well, one area that holds tremendous promise is personalized medicine. Imagine being able to tailor treatments to an individual's unique gene expression profile. So instead of a one-size-fits-all approach, we could have treatments that are specifically designed for each patient.
So, like, instead of just treating a disease, we could be treating your disease based on what's happening at a cellular level in your body.
Exactly, and we're already starting to see this happen in cancer treatment, for example. Some cancer drugs are specifically designed to target proteins that are overexpressed in certain types of cancer cells. So by understanding which genes are turned on or off in a particular patient's cancer cells, doctors can choose the most effective treatment. And as we learn more about gene expression, we'll be able to develop even more targeted and personalized therapies for all sorts of diseases.
That's amazing. It seems like we're really moving towards a future of personalized medicine, but are there applications beyond just treating disease? Could we use this knowledge to improve our lives in other ways?
Oh, absolutely. Gene expression analysis is being used in all sorts of fields, from agriculture to forensics. For example, scientists are using gene expression data to develop crops that are more resistant to pests and diseases. And in forensics, they're using it to identify individuals from DNA samples.
Wow, it's amazing to see how this knowledge is being applied in so many different areas. So big picture, what's the most important takeaway for our listeners? What should they remember about gene expression?
I think the key message is that our genes are not just static blueprints. They're dynamic and responsive to their environment. Our experiences, the food we eat, even our thoughts and feelings, all these things can influence which genes are turned on or off. And it's this intricate dance of gene expression that ultimately shapes who we are.
That's a really powerful thought. We're not just passive bystanders, we're active participants in the expression of our own genes. And the more we understand about this process, the more we can take control of our own health and well-being.
Exactly. It's a whole new way of thinking about ourselves and our relationship with the world around us. And it all starts with understanding this incredible language of life, this code that's written within each of our cells.
So much information encoded in our genes. It's incredible. And with these techniques, it's like we're finally starting to crack the code.
Yeah.
Understand the language of life itself.
Yeah. It really is remarkable. The more we learn, the more we realize just how complex and interconnected everything is.
I'm curious, though, like, are there any real-world examples of how this knowledge is being applied right now?
Oh, tons of examples. Like in cancer research, gene expression profiling is already being used to help doctors identify specific subtypes of cancer.
So it's leading to more targeted treatments.
Exactly. By understanding which genes are turned on or off in a patient's cancer cells, doctors can choose the most effective therapy for that individual. And in infectious disease research, gene expression analysis is helping us understand how pathogens interact with our cells and how our immune system responds to those infections.
So we're learning how to fight disease more effectively? Are there other applications?
Absolutely. For example, in agriculture, gene expression analysis is being used to develop crops that are more resistant to pests and diseases.
It really seems like the potential applications are, well, limitless.
They really are.
Yeah.
As the technology keeps advancing, we can expect even more exciting breakthroughs in the years to come.
Okay, so zooming out for a second, what's the big takeaway for our listeners? What's the one thing they should remember about gene expression?
I think the key message is that our genes are not just these fixed, unchanging blueprints. They're dynamic. They're responsive to the environment, Our experiences, our diet, even our thoughts and emotions, all of these things can influence which genes are turned on or off. And this incredibly complex dance of gene expression, that's what ultimately shapes who we are and how we function.
It's almost like a conversation. Our genes are in constant dialogue with the world around us.
Exactly.
Well, this has been an absolutely fascinating exploration into the world of gene expression. I have a feeling our listeners are going to be looking at themselves, at life itself, in a whole new way after this.
I hope so. It's really a reminder that there is a whole universe of activity happening within each of us. And honestly, it's pretty amazing, the intricate mechanisms that make life possible. It's mind-blowing when you really stop to think about it.
And on that note, we'll leave you with this final thought. If our gene expression is constantly changing, always adapting to our experiences and our environment, what does that tell us about the relationship between nature and nurture? How much of who we are is predetermined by our genes, and how much is shaped by the world around us? Something to ponder. Keep those questions in mind and keep exploring. There's always more to discover.